Just very quickly, I wanted to, I mentioned to you the other day that article about Romney making a policy statement about embryonic stem cell research and said that would, here was sort of examples of issues we were thinking about in this course that would come into your ordinary life. Here's on today's Globe. There's a Kennedy rips Romney over stem cell policy. This debate is continuing. It's on the front page of today's Boston Globe. We have two people who probably, neither of whom probably have the background in biology that you guys are going to have by the time you finish this course, having to grapple with these very serious scientific issues that have all kinds of other implications. Here's another thing. This was in yesterday's newspaper. This was on, I think, the second page of the Boston Globe. It's about a curator over at Harvard at the Museum of Comparative Zoology. They've got all these different birds, many, some of whom are extinct and something. And now the curator, instead of just having them there as a sort of collection, and that's all it is going and getting DNA out of these specimens and working back and looking at gaining new insights from birds that don't even, animals that aren't even found on this planet anymore. And this one was from, I didn't want to show you this on Valentine's Day particularly, but this is critical. I don't subscribe to the New York Times, but for some reason one appeared on my porch on Saturday morning. I opened up the first page, rare and aggressive HIV reported in New York. I'll be talking to you about the Hiv-1 virus, which is the virus that ends up destroying some of your major defender cells in the immune system, and then people die of AIDS, which is sort of all the other stuff that then happens. And it was terribly scary when it first showed up. There was no treatment for it. It's like that in most of the third world, and people die if they get it. I lost a friend to it, a close friend to it, after it first came up. Then they came up with these various cocktail of different inhibitors, and the idea was, instead of just having a single drug target where it would be easy for the virus to mutate, and as I'll tell you, the HIV virus mutates like crazy. Instead, they'd use multiple targets, and that's why they do it. And sort of then the chances of getting a mutation is you multiply all these low probabilities of getting a mutation, and you would hope it would never happen. It's happened. There in New York is a version of HIV-1 that is resistant to everything. If that spreads, which it probably will, it'll be just like what happens with all the bacteria and antibiotics. It works for a little while. Natural mutation and natural selection, something will happen. The organism or the virus will change, so it's no longer resistant. So, for the moment, as always, make sure you never expose yourself to AIDS. This would take us back just like to before any of these things were inhibited. And the same thing goes right now. There's strains of Mycobacterium tuberculosis that are resistant to every known antibiotic. If you get tuberculosis with that thing, it's like you were living 200 years ago. They can't do anything for you. They put you in a sanatorium, but you can't be cured. This is worse. This one kills, this is awful. This one kills you. So just be careful. This is just the tip. But what always happens when you have antibiotics or drug treatments, eventually natural selection mutation will probably produce a variant. And this looks like it's happening. I don't know, but I hope not, but I'm afraid this won't be the end of this one. Okay, so some of you, I've read some of the comments. I'm not terribly surprised that some of you think, why is he telling me all this? God, there was a lot of work for tremendously confusing. There were all these chemical structures. I didn't really get it all. It seemed that somehow out of this, the cell got 2 molecules of ATP. Why are we wasting all the time? Why am I wasting all this time telling you about how to get 2 molecules of ATP? But I mean, this is basically what we were doing. We were looking at glucose, which is C6H12O6, going through this process, which pathway of 10 enzymatic steps, and what ends up coming out at the other end is 2 pyruvates, and the net yield of that is 2 ATP plus 2 molecules of this weird thing called NADH. Now, I think most of you, although a few of you still don't seem to have gotten, the reason you need to make ATP out of ADP and inorganic phosphate is it stores energy. It's, like having electricity in your house or batteries in your flashlight or in your iPod or whatever it is. You got to have energy in order to do stuff, and cells need it. And the major sort of energy currency they use is ATP, and they put most of it by squeezing together a phosphate up to an ATP, making this bond it's stable unless there's an enzyme that'll break it open, but there's energy stored in it, and they'll drop downhill, and you can use that energy to drive other reactions. So for life to happen, it had to somehow figure out how to get energy in a form that it could use to do all the biosynthesis that's necessary for life to go on. NADH, I showed you the molecule. You've got a structure. The cell makes some number of molecules of that. It's around. And what happened, it comes in, it helps some of these processes go. It ends up, it's sort of like a banking system for electrons. If it gets NADH, it's got electrons stuck on it. There's only a limited number of molecules of NADH in a cell. So if you put you make all the NAD molecules into NADH, then you have nowhere else to put electrons, and everything comes to a grinding halt. Because I was talking about redox reactions. If you're taking electrons off somewhere, they got to go somewhere else. And the cell uses NADH as kind of almost a currency for passing electrons around in just the same way it uses ATP as a currency for carrying energy around from one form or another. Now, the problem was, and why this was so important, is this apparently, we're basically looking at an evolutionary fossil of some sort, because every organism on Earth uses, or virtually every organism on Earth uses glycolysis. It's in our cytoplasm of our cells, it's in the cytoplasm of bacteria, it's in the cytoplasm of archaea. It apparently evolved so long ago that every form of life on Earth, depends on this. Anything that can metabolize a sugar molecule, pretty much. And so it looks complicated. It is. Maybe you could have designed, I'd like to tell you that it's 2 reactions and you make 2 ATPs and it's all simple. Life isn't often simple. It's often very complicated. It wasn't done by a team of design engineers in Building 10 designing it. It was done by something that happened with random selection. When something finally worked, apparently it got fixed in evolution. And even if it looks clunky, I think the other way to look at it is to say, Mike, isn't that almost I feel a sort of sense of wonder at this point. And granted, I've had a lot of years of working in the field, that something that complicated could work out and take this molecule of glucose and make it into 2 molecules of ATP that can then be used to allow the cell to do synthesis. As I'll show you, there's a better way you can use this much more efficiently, 18 times more efficiently if we do respiration, which we'll talk about today, but that needs oxygen. And when life started 3.8 billion years ago or so, there was no oxygen. So it wasn't an option. The initial organisms had to make do with what they found, and this is what developed. Now, the problem here, so ATP can go off and be used for lots of things. The problem, if there's no oxygen around, as I said, there's a limited number of molecules of ATP. So to make that glycolysis go, you're taking some electrons off and doing that oxidation steps, you're reducing NADH. And if you want that to go as a cycle, you've got to somehow regenerate the NAD+. So you can go through another cycle and make more ATP. So there were 2 variants that I told you about. If you don't have oxygen around, one of these variants yields 2-lactate. And so the net yield of this is 2 ATP. Because what happens in this part is it uses up 2 molecules of NADH, changing the 2 molecules of pyruvate to 2 molecules of lactate. So that gets rid of this, and the net yield of the whole thing is 2 molecules of ATP. The other way to do it is the way yeast does it. It makes 2 molecules of CO2 plus 2 molecules of ethanol. And I think when I wrote that on the board, I forgot I showed you that there was acetaldehyde. We had 2 molecules of that going to 2 molecules of ethanol. And I think I forgot to write on the board that it used up 2 molecules of NADH, because that's important. So that's what this whole thing is about. It achieves the same thing, and the net yield of this one is 2 ATPs as well. So I'm going to talk about photosynthesis later at the latter part of the lecture, but if you maybe recall, the initial sort of thing I called release 1 came up here about 3.4 billion years ago. Later on, an improved version came along that started to generate oxygen. Now, it took billions of years for us to get to our present level of oxygen. But when oxygen became available in the atmosphere, then it was, there was a new option for making energy. And that was, instead of taking this NADH, and remember, there's about 50 kcals per mole of energy in there, it's just being thrown away. It's not being used at all. But if there's oxygen around, there's a new and better system, which you know is respiration. It's a word you've heard. You know, we breathe oxygen. We need to breathe oxygen because we're using the oxygen to make energy. And I'll tell you again, you don't have to know all the details, but there's a biochemical cycle called the citric acid cycle. And it goes together with another process that's known as oxidative phosphorylation. And so this is, if we have oxygen, what we end up with at the end of the day, instead of these products, is 6 molecules of CO2 plus 6 molecules of water, but more importantly, 36 molecules of ATP. So respiration is tremendously better at capturing energy from a glucose molecule, but this is, I'll show you is a later arriving development in evolution. It had to be because it required oxygen in the atmosphere, and so even though we only get 2 molecules out of ATP, we all do it. And the other thing it generates is pyruvate, and as you'll see, this process takes those pyruvates. That's the starting material for this part over here. Okay? I hope this is making a bit more sense as to why you've got to keep your eye on the big picture, or all you're going to see is a whole lot of structures and chemical transformations that don't make any sense. This is all about making any ATP and energy, and as we'll see later on, photosynthesis in cases that can be something to do with making reducing power for biosynthesis. So in order to understand this, though, I have to introduce you to another way of thinking about energy. Some of you still seem to be struggling with the idea that you can store energy in a chemical bond, but I think from the comments, the majority of you have that. If you're having trouble, ask your TA or look in sections and stuff. But the insight to how this part came, even though this process, what underlies this was invented about roughly 3.4 billion years ago, scientists didn't begin to even get a glimmer of how this worked until about 1961, when there was a scientist named Peter Mitchell who got a Nobel Prize for the insight that he had. And what he recognized, that there's sort of 3 forms of energy that are interconvertible. The energy of a chemical bond, and I keep telling you this, that ATP, if we hydrolyze that, it goes to ATP plus inorganic phosphate. The delta G prime 0 is about -7 kilocalories per mole, but under physiological conditions, it works out that each ATP gets you about 12 kilocalories per mole, because life doesn't happen under these standard conditions. So that's one form of energy. We've been talking a lot about it. There's another form of energy which you probably know intuitively, and that is if you have a high concentration of some compound on a side of a sort of impermeable barrier and a low concentration on the other side, high concentration of sugar, low concentration of sugar. If you give the system a chance, it'll come to equilibrium. So the concentrated stuff will flow downhill until you're concentrated on both sides. There's energy you could get out of that. So there's energy basically stored in a concentration gradient. There's another form which you'll probably be familiar, certainly to some of you, and that's the idea you can store energy in an electrical gradient. If you have some kind of impermeable barrier, and we have a lot of charges on one side and less charges on the other side, then there's a polarity, there's an electrical gradient, and that's a form of stored energy. Now, it happens that a membrane is impermeable, as I've told you, to most things. So you can get a high concentration of something on one side and a low concentration of the other, and control by a protein embedded in it whether those ever get a chance to come across. The same idea applies for an electrical gradient, and in particular of interest to biology are hydrogen ions. So if we have a situation like this, where we have more hydrogen ions on one side of a membrane than another, then we have an electrical gradient, we have a polarity. And so this is the membrane. And so all cells then have and this is the way it works is this is the outside of the cell and this is the inside of the cell. So more pluses on the outside, hydrogen ions on the outside than the inside. And it's about 70 millivolts. May not sound that impressive, right? Another way to look at it, though, is the membrane is about 3 nanometers. So if you say, well, okay, what's the gradient of electrical gradient across that? It's about 200,000 volts per centimeter. And high tension lines are, more like that per mile or something like that. So although it seems modest because the membranes, you get a very, very powerful electrical gradient in all cells. And so this is a form of energy. And I think this was proposed in 1961. Textbooks often say it was adopted in the early 70s. I went to a postdoc at Berkeley in 1975, and people were still having arguments about whether this really was the way, was this really something that was used in nature. It is, and I think one of the most dramatic demonstrations that a proton gradient can be a source of energy comes from this sort of thing. I showed you how these bacteria, these are E. coli swimming around with these rotary motors that spin 10 to 100,000 RPM, driving those flagella. I showed you the picture of the inner membrane of E. coli, and I mentioned it has a double membrane. This is a sort of a protective layer. You'll see a little bit later in the lecture some double membranes. Again, and here's the motor with the big propeller, the flagella sticking out from it. And the way this thing, and I showed you, I guess, this picture and then this textbook diagram, what drives this motor is our protons flowing from the outside of the cell through here, through the proteins in here, through channels in them, and that's what provides the torque for the motor. That's what drives it. It's not ATP or anything. It's protons from the outside to the inside. And there's a very dramatic demonstration of sort of like, Friday night horror films, if you wanted, in some way where people found a way of popping an E. coli open, so like all of the cytoplasm ran out, and then it would reseal. So what you've got is sort of an E. coli that is just a shell, just the membranes and the proteins embedded in it, and it just sits there at neutral pH. If you now acidify the medium, what's happening? You've created a proton gradient because there are now more protons on the outside. And this is just the same picture I showed you before. But what happens if you do that experiment is the bacteria start swimming. They don't have any insides or anything, but you have created artificially a proton gradient, and it drives the motor, and they start swimming. It's sort of like, dead men walking or something at a bacterial level. I think it's a really dramatic demonstration of how you can use the energy of a proton gradient to make energy. So the principle that underlies how respiration works and photosynthesis works is that you take advantage of this combination of concentration and electrical gradient, and it's known as the chemia's osmotic hypothesis, because you have an electrical gradient because of the charges. You also have a concentration gradient because you got more hydrogen ions on this side, so you can't really separate them, and they're kind of coupled. But this idea is that life uses this proton gradient in order to make energy and do some of these energy transactions. So here's the sort of principle of how it's done, is you have a membrane like this, then we have a protein, and now we're not going to see all the alpha helices and beta sheets. It'll be one of those things we talked about that spans the membrane. The membrane, as you might guess, from what you know about it, is impermeable to hydrogen ions. So that what this protein does, that's embedded in the membrane, it's a proton pump. And if you provide it with energy in some form, what it does is it takes a hydrogen ion that's on the inside and it makes it into a hydrogen ion on the outside. There's no chemical transformation of the proton. It's just gone from one side of the membrane to the other. It's almost like recharging a battery or something, if you want to think about it, perhaps in that kind of way. And that's then the second stage is once the proton gradient is established and you have now many more H pluses on the outside than on the inside, then there's another protein that lets the proton flow. The proton flows down the gradient and therefore is able to come back inside the cell. Now, if that's all there was to it, we wouldn't have achieved anything. We would have wasted energy, pumped something out, pumped something back. But this molecule has an interesting property, and that is the ability of the proton to flow down the gradient is obligatorily requires ADP plus inorganic phosphate. And as the proton comes down this energy gradient, there's enough energy given off by that the cell is able to capture it and use that energy to synthesize a molecule of ATP. Got it? So proton you do some energy, like from light and photosynthesis, or we'll see in a minute, other ways of doing it, and then get it outside. Once you've got the gradient, now you can make ATP. And actually, one of the completely remarkable discoveries of structural biology, this is known as an ATP synthase. It's a protein. It's an enzyme. It's those kinds of things we've been talking about. This is also a protein. You see all the different things proteins do. So this ATP synthase, which uses this energy of the proton gradient to make ATP, its structure's been worked out. And at a level, here's part of the crystal structure. You can probably see some alpha helices, beta sheets. Here's a textbook diagram of it showing it's upside down from them, showing you, but here's a proton on the inside flowing across. And what's remarkable is it turns out that this ATP synthase is structurally related to the protein that's at the heart of that rotary motor that drives the flagella. And in fact, remember, I think I showed you where you could stick the flagella to a cover slip and the bacteria twirled around, so you could actually see they were rotating. So people did sort of the equivalent thing. They managed to stick this ATP synthase, and then in order to see, you couldn't see that the top part was turning, but they used some tricky stuff we'll talk about with antibodies and something else to attach a long filamentous molecule. It's a polymer of protein called actin. That's the same stuff we find in our muscles. And it made it long enough you could see that when this thing was working, that it was twirling around. And so evolution took this same basic sort of piece of protein machinery, and in one case it used it to capture the energy of a proton gradient and make ATP, and in another case it used it to drive this propeller, if you will. And here's a sort of a simple sort of diagram. So as the proton flows, kind of what happens is the inner part of this thing sort of goes click, click. And every time it does it, it synthesizes an ATP, and it sort of takes that energy to push together the ADP and the inorganic phosphate, overcoming that activation energy and getting them close enough that you're able to get that bond. It's a truly remarkable thing. This is a somewhat hard concept to grasp, I understand, but if you can understand this interconvertibility between energy in the form of a combined concentration and electrical gradient and ATP, and nature goes back and forth and back and forth, and it's absolutely fundamental to life. If we didn't have this stuff going on, we couldn't do it. You know, as I say with glycolysis, I wish it was simpler, but this is the way nature did it. This is the way we are, and if you want to know about biology, this is how it goes. And I wouldn't be doing you justice if I didn't tell you some of the details. You're MIT students. You should be able to handle, I hope, some of how the world actually works at this kind of level. Okay, now, with that kind of background, now I think we can kind of, pretty quickly, I can help you begin to see what happens here. Now, remember the problem up there with glycolysis. It started when there was no oxygen, and therefore it generated these NADHs that weren't any good. You had to just get rid of them, so you took the pyruvate organisms, learned how to put them to make lactate or ethanol and carbon dioxide. But if there's oxygen around, then there's another possibility, and that is you can combine these molecules with oxygen. So if we take 2 NADH plus 2 hydrogen ions plus a molecule of oxygen, what we get out is 2 waters. And I think I can show you what's going on there kind of simply. What NADH was, we got a pair of electrons here and a hydrogen ion. Well, if we took that, what's that? I think you'd recognize it as a molecule of hydrogen gas. So if you take NADH and oxygen, what the cell is really essentially doing, it's taking hydrogen gas plus oxygen, and it's giving 2 waters. So it's basically burning hydrogen. And I think most of you know what would happen if I had a mixture of hydrogen and oxygen up here and chucked a match at it or something. We'd have a massive explosion. And in fact, that's why there's 50 kilocalories of energy released when that happens. And so on an energy sort of diagram, these free energy diagrams, we had the 2 NADH here plus the molecule of oxygen, and down here we have the 2 molecules of water. There is this, if that happened in one step, it'd be a huge amount of energy. No cell or organism in Earth has figured out how to do that in one step, and I think some of the textbooks liken it to sort of, it would be like setting a stick of dynamite off in the cell. And this is where one of these things that seemed probably like a kind of uninteresting thermodynamic property to some extent becomes to be really important in understanding biology. And that is the fact that, remember I said this drop in energy from reactants to products was a thermodynamic property. It didn't matter whether like you skied straight down the hill or you came down in a series of little things, you still got the same amount of energy released going from here down to there. So that's in essence what the cell does, is it breaks this, takes this energy and it breaks it into little packages that it's able to manage in a chemical way. And so instead of coming down at once, it comes down in a series of steps, and every time it flows partway downhill, it does something. And what it does is it passes 2 electrons to some kind of carrier, and then the 2 electrons flow downhill a little bit to another one, and then to another one. And what happens when these electrons are flowing downhill, though, is that they drive that proton pump. So a proton pump takes the proton from being on the outside to the inside, and when the 2 electrons drop down to the next level, another H plus goes from out to H plus in, again here. And if you understood what I said before, what the cell can now do is it can make 3 ATPs by using that ATP synthesis, and now taking advantage of those 3' proteins and making them into 3 ATPs. And then at the end of the day, what happens then is these 2 electrons get together with 2 hydrogen ions plus, I'll show it as a half of an oxygen here, to give you water. And what's happening up here is, in essence, the molecule of sugar, C6H12O6, is being burned with 6 molecules of water plus this to give you 6 molecules of CO2 plus 6 molecules of ATP. So the cell is essentially achieving the same kind of thing by this process as if it had burned it, you'd burned it in oxygen. There's that much potential energy released. And in order to, the total amount of this change, the D prime 0 is something of the order, I think it's minus 686 kilocalories per mole. It's able to capture respiration, captures about 60% of that energy as ATP. This process of fermentation, this is what these processes that happen in the absence of oxygen are known as fermentation. As you can see, they're much less efficient. It gets more like 3.3% of the energy captured as ATP. So you can see when oxygen arose in the environment what a huge boon it was to life, because you could get a lot more energy for the same amount of starting material. There is one thing, though, that the cell has to do. In order to do this, it has to do some more chemistry, because it has to take those pyruvates and it has to somehow run them through this thing that I'm calling the citric acid cycle and oxidative phosphorylation. Well, the oxidative phosphorylation is basically this chain I've diagrammed for you here in a schematic way, and that's about the level you'll have to understand it. I mean, physically, it's going to be a bunch of proteins stuck in the membrane, and as the electrons get passed along, they pump a proton as the procedure this pathway occurs. But the other thing the cells have to do is they have to take those two pyruvates and they have to burn them all the way down to CO2 and water. And what's happening, if you remember that chain of oxidations, the carbons are being successively oxidized all the way up to carbon dioxide. You can't be any more oxidized than that if you're carbon. What that must mean is something else is getting reduced. And what gets reduced, where the electrons go, is NADHs. So once oxygen became available in the atmosphere, then the name of the game was to take those two pyruvates and to somehow burn them all the way down to here and therefore generate as much NADH as possible. And if you can make some ATPs along the way, well and good. So there had to be a whole other set of chemical reactions. It's every bit as complicated as glycolysis that emerged in nature that carried out that job. And this time I'm not going to take you through all of the chemical structures. You can look at it in your textbook and stuff, but what I really want you to kind of take is to watch, keep your eye on the number of carbons. If you look at the structure of pyruvate, you'll see that it's three carbons. It was this. There's a keto group and an acidic group. So this is almost carbon dioxide. It's just one oxidative step away. So what happens in this cycle is that first the carbon dioxide is removed, and this makes an acetate. In essence, it's actually joined to something right here. And then this feeds into this thing called the citric acid cycle. And what it is, as I said, is it's a set of chemical reactions that are designed to take this pyruvate and burn it all the way to carbon dioxide and water, and to generate as many NADHs and ATPs as it can along the way. And again, I wish it was simpler. It would be really nice if it just did it straight from acetate. But instead, you'll see if you look at the Tca cycle, there's the C4 compound, and this gets joined together to give a C6 compound. Then it oxidizes one of these carbons to give a carbon dioxide. Now you're down to C5. It does it again. Another molecule of CO2. You're at C4. And then it takes this C4 carbon skeleton, puts it through some transformations that enables the cycle to go all over again. So basically, this C4 thing just goes through the cycle. It's a carrier, takes this C2 right here, lets it go be processed to give CO2, 2 molecules of CO2. In that process, it generates more NADH, a bit of ATP, and one other kind of reduced carrier that you can think of for the moment, largely equivalently to NADH. Okay, now the entity in our cells that carries out this process of respiration and of respiration, so the citric acid cycle and the mitochondria, is not actually in our cytoplasm, which is perhaps surprising, but all the enzymes for glycolysis are. So remember my simple diagram of the eukaryotic cell. On the first day, we had the nucleus, which is a membrane compartment that has the DNA inside it. We'll have more to say about that in the next lecture or so. But then there were some organelles, and one of them I said was a mitochondrion. And I also said in the first lecture that there's pretty good evidence now that the mitochondrion arose by some earlier progenitor or ancestor of a eukaryotic cell capturing some kind of bacterium. It's actually one that's sort of kind of related to E. coli, had double membranes. And the mitochondria actually still have some of their own DNA. But this is where all the energy, all the enzymes for the citric acid cycle and oxidative phosphorylation are found in the inside of the mitochondrion, inside of something that probably used to be a free-living bacterium. In contrast, the enzymes for glycolysis are in what's called the cytoplasm of the cell, which is sort of the main part of the insides of the cell. So you see even here this sort of evolutionary history that I kind of put out across the board. The enzymes for glycolysis arose so early in evolution, they're in the cytoplasm of virtually every organism on Earth. Eukaryotic cells managed to figure out how to get 18 times more energy for a molecule of sugar. But in order to do it, they kind of had to cheat. They took a bacterium that figured out how to do it, put it inside our own cells, and then it's able to run now. It's become a part of our cell, and it's where all this process takes place. And if you look at the structure of a mitochondrion, it's actually got 2 membranes. You just saw a picture of that in E. coli. And in fact, they're sort of kind of involuted a little bit like this. And so this is an outer membrane of a mitochondrion. So I'm basically taking this and blowing it up. So this is a mitochondrion. And the in and the out, in this case, there's an inner membrane. This is where the proton pump is located. This is where the ATP synthesis is located. And when a mitochondrion is working, what it's doing is it's pumping hydrogen ions from its inside, which is more or less the equivalent of its cytoplasm. It's whatever used to be the cytoplasm of the original bacterium, outside into this space between the inner and outer membranes, and then it generates ATP by flowing back down. Okay, so there are a couple of sort of things that affect your life that come out of this. One of them is understanding the freshman 5 or freshman 15 or whatever it is. You come to college and all of a sudden you put on a lot of weight, and sometimes you can figure it out. It's because you ate a lot of Ben & Jerry's or a lot of chocolate bars or something and didn't do as much exercise. Another one of you said, gee, now I understand why I'm not putting on any weight. I do water polo, and I guess I must be burning up everything I eat as energy. And you're right. But you can now begin to, I can show you at a molecular level what's happening. So what happens if your body is, everything's regulated, and it can tell if it needs, cells in your body can tell if it needs energy. If it needs energy and you eat sugar, it runs right through here and it makes ATP. But if you've eaten enough, and the things that monitor your body say that you've got enough ATP, then it doesn't keep this process going. Instead, what it does is it stops it, and it says, instead, we're going to put something away for a rainy day, which you could see would make a lot of sense in evolution. You know, you want to, if things are good, you just kill the mammoth and you can all eat well for a while or something. It would make sense for your body to be able to pack stuff away. So what it does is it intercepts the process at this level, at this C2 level. The pyruvate gets to here, and instead of being run through this in ATP, this acetate or acetyl moiety, which is a C2, gets run through a cycle of successive 2-carbon additions. And what comes out of that are fatty acids. It also takes 3-phospho glyceraldehyde. You might remember that. That's one of the products stuck in the middle of that glycolytic pathway. And it makes that into, this is a 3-carbon compound. It makes it into glycerol, which is a 3-carbon compound. And if you look back when we talked about lipids a couple of lectures ago, you realize what happens if you combine a fatty acids with glycerol, then you've got fats. And in essence, I mean, that's what happens. We put on weight if we eat more than we're burning. Our bodies say, okay, I've got as much ATP as we need. I'm going to take some of that ATP and use it in this kind of way. Just a sec here, let me just see. So there's another thing that happens when you're physiological thing that we experience. Well, we seem to be stuck at the moment. I won't worry about it. I've run some marathons. I don't know, a few of you may have. If not, almost all of you have heard about hitting the wall. And it happens in general around 21, 22, 23 miles. Depends on condition you're doing. And physiologically, I have experience. This is amazing. I understand why they call it hitting the wall. You're running along. You think, boy, I'm tired, but I'm doing okay. And in the space of a quarter mile, it's like you banged into a brick wall, and you can sort of keep yourself going. But it's a profound physiological change. And what has happened at that point is you've run out of sugar to burn. Now, I've told you you can take sugars and you can polymerize them into glycogen. That's one of those polymers with alpha-1,4 linkages. That's a sugar storage molecule. So if you start running, you carbo load, you try and get as much sugar into glycogen as you possibly can. You start running the marathon, your body starts taking that glycogen and breaking it down into sugars. It runs it through that process. What happens when you hit the wall as a human body is most human bodies are designed so that you run out of sugar. You just can't carbo load enough to do 26 miles. You can get through 20 or something there. And when you run out of that, what happens then is your body can no longer burn sugar. So it switched to burning fatty acids. And that's less efficient, and you really feel it. If any of you have done long endurance things or triathlons or anything, you've experienced that change. The final thing, just to close the lecture for today, is one thing you can appreciate. Yeast is somebody that can do both of these things, right? It can either grow anaerobically, and it makes CO2 plus ethanol, or it can grow aerobically, and this gives it 2 ATPs per glucose, and this gives it 36 ATPs per glucose. So if you were a yeast, you would have to somehow regulate the rate of glycolysis depending on whether there was oxygen around or not. And it's very tricky, the neat way it happens. There's an enzyme, that wouldn't surprise you, that has an active site for one of the key steps in the pathway, where it's going from fructose 6-phosphate to fructose 1,6-diphosphate. That's one of the intermediate steps in glycolysis. It's the enzyme that turns out to be rate limiting. You can control passage of something through a pathway by just making one step rate limiting. So this has a place for the fructose 6-phosphate plus ATP to bind, and it catalyzes the formation of the fructose 1,6 diphosphate. But that enzyme has to work at different rates depending on whether the cell is aerobic or anaerobic. So what it does is there's a second binding site over here, and if it binds ATP, this speeds up, slows down the rate that's catalyzed over here, and that's what you'd expect. If it's got enough ATP, it doesn't need to run glycolysis as fast. And if it also binds AMP or ADP, and that speeds up the rate. So the yeast are able to monitor, to sort of have a control on how fast sugar flows through this glycolytic pathway. And what they're really doing is they're monitoring, do I have enough ATP or not? And so if they're running with respiration, making lots of stuff, they don't need to do glycolysis as fast. If they're anaerobic, they have to run it 18 times as fast to get the same amount of energy. Okay? We'll begin the next lecture with photosynthesis, and then we're going to get into a bunch of molecular biology. Okay?